To the chairperson, Professor Ramakrishna and Professor Shinjini Bhatnagar, to chair this session. And I am going to take you from the dietary habits that has been talked to, fermented food that has been talked to. I am taking you to a different story of comparative assessment of the predominating gut flora during inflammatory bowel disease. Now, as we know that this inflammatory bowel disease is an autoimmune disease, and there is no pathogenic organism has been found so far to be associated with the disease. And it is a multifactorial disease. It has a genetic component, and it also has a microbial component which exaggerates the immune response. So far this is the story that we know about the inflammatory bowel disease, which involves two diseases, ulcerative colitis and Crohn's disease. And the study that we have done in our laboratory mainly concentrates with the ulcerative colitis patients, which we have collected the samples from the gastroenterology department of All India Institute of Medical Science with my collaborator, Dr. Vineet Ahuja. Now, coming to the contributions of the microbiota to the host, which has been deliberated in detail in the morning session as well as in the afternoon session. And what we see that the digestion and the fermentation of the carbohydrates is very important contribution that the microbiota makes, and it also involves in the production of vitamins, development of the GALT, that is the gut-associated lymphoid tissue, polarization of the gut-specific immune responses, and the prevention and colonization by pathogens. And in turn, the gut immune responses that are induced by commensal populations regulate the composition of the microbiota to maintain the homeostasis. Now, a paper which appeared in Nature Review Immunology in 2013 says that the gut plays a protective as well as a pathogenic role. Now, in a normal individual, we find that the gut microbiota is in a symbiotic relationship, and the types of bacteria that are present so far, of course, the genome projects have come and microbiome projects have come, but still there are many bacteria which still remains unclassified. So there are certain unclassified bacteria, there are certain beneficial bacteria, and there are certain pathobiomes. Now, the purpose of these pathobiomes are that the useful bacteria keep the pathobiomes population down, so that the gall development and the Th17 cells, Th1 cells, which are responsible for the production of the pro and anti-inflammatory cytokines, can take place, and it also helps in the barrier function, Reg3 generations, etc. And it also plays an important role in the production of Treg cells and the anti-inflammatory IL-10 cytokine. And these symbionts also keep, in the due course, they keep the pathobionts in control. But when a dysbiosis takes place, or when there is a disease and there is an alteration in the gut flora, it seems that the dysbiosis, because of the dysbiosis, there could be a production of the pathobionts, which are not coming from outside. They are indigenous flora. But some of these pathobionts might dominate during the disease condition. And because of this, it disrupts the microbial community structure, and there is an accumulation of cholitogenic pathobionts. The pathogenesis of IBD is mainly located in, if you look at the pathogenesis, we find that it is located in the areas with high bacterial concentration. As I told you, there are two diseases. In case of Crohn's disease, it is mostly located in the terminal ileum and cecum, and in case of ulcerative colitis, it's the rectum area. The central event in the pathogenesis of IBD is the exaggerated immune response to the commensal intestinal bacteria or to its specific members. And this idea came because in a genetically modified animal model of IBD, do not develop colitis under germ-free conditions. So this clearly shows that there is a huge role of the microflora or the indigenous microflora in the manifestation of this disease. Now, we started our work somewhere around 2008, And that time we are working on the mucosal flora of the biopsy samples that we took from the hospital. And our results had shown clear delineation of the concentration of the mucosal flora between the predominating and subdominating genera under disease conditions. And in the previous speaker has already pointed out which are the dominant and subdominant flora in the gut, indicating that the subsets of bacteria participating in the pathogenesis of ulcerative colitis and Crohn's disease are likely to be different. And in the present study, what I am going to present today is to show, to evaluate and compare the changes in the diversity of fecal flora in the Indian patients. Right now, we are working on the fecal flora of these patients from healthy to disease state during ulcerative colitis, and also quantified the bacterial population using certain specific probes that we have generated in our lab in control versus patient samples. And in the patient samples, we have taken 3 categories of samples: moderate, severe, and remission stages of the disease. And we tried to obtain any significant association of the bacterial flora during the disease condition. Now, these are the dominant and the subdominant bacteria of the human gut, which we have studied in our samples, that is Bacteroides, Bifidobacteria, Clostridium coccoides members, members of Clostridium leptum, Enterobacteriaceae members, Lactobacillus, Enterococcus, Atopoboium, Methanogens, and sulfate-reducing bacteria. And the techniques that we have used to illustrate these changes or the fluctuations in the gut flora involves fish microscopy, fish flow cytometry, and qPCR analysis. And we have also looked into the metabolites, the changes in the metabolites by GC analysis. And we have tried to do a preliminary study, this is still an incomplete study, to figure out the important changes that have taken place at the species level by using a denaturing page. And the probes that we have used for the flow cytometry are the EUV probe, that is positive control probe, for the to measure the total bacteria, which is leveled with FITC, and a non-EUV 338, which is to see if there is any background signal, that is with non-EUV 338, which should not amplify any of the bacterial species. And the genus specific probes that we have used were leveled with Psi5 at the 5' end. So this is the quantification of the Bacteroides prevetola group. Here we show the fish microscopy of whole cell in situ hybridization of the straight from the fecal sample with genus specific Psi5 leveled probe. So this is for the Bacteroides prevetola group, and down you can see the fish flow data which we have generated. And if you look at the graph, you can see that there is a control, there is a moderate, there is a severe, and the remission cases. And if you see carefully, you will see that there is a relationship when we compare the control, the severe and the remission. We see the fecal flora coming down, both by flow cytometry as well as the qPCR analysis of this Bacteroides flora. And this Bacteroides actually goes significantly high in the severe patients if we compare it with the control individuals. So the population of Bacteroides increases significantly during severe condition and reverts back to normal during remission. Now, when we quantified the genus Bifidobacteria, again with the flow cytometry, we find that there is a significant reduction which is expected in the Bifido population during the disease condition. But what we find that in the remission patients, again the flora comes back towards the normal level. And this was proved both by flow cytometry as well as the quantitative PCR analysis. And on the upper panel, we show the bacteria that are stained with the Psi5 leveled Bifidobacterium probe. So the population of the Bifidobacteria drops significantly during severe condition and reverts back during the remission stage. Coming to the Clostridium coccoides group, again we find that there is a significant drop in the moderate followed by the severe condition of the disease, and the patients which are in the remission stage again seems to revert back to the normal level of Clostridium coccoides, which is proved again by flow cytometry and the qPCR analysis. The members of Clostridium cocoides drop significantly during moderate and severe condition, but reverts back during the remission stage. In case of Clostridium leptum, it was very interesting that we found that there was a drop of members of Clostridium leptum group, and there is a drop in the moderate as well as in the severe cases. But in the remission cases, we did not find a significant turnover or significant reversion back to the normal level, both by flow cytometry as well as by qPCR. So this shows that the members of the Clostridium leptum group, which actually comes down, they do not revert back so soon. It takes time probably. So the population of Clostridium drops significantly during disease condition, but do not revert back during remission stage of the disease. So when we saw that there was a significant reduction in the butyrate producing bacteria, that is the members of the Clostridium group, we went a little bit in detail to design primers and the probes for Faecalibacterium plasmodium, Roseveria intestinalis, and Eubacterium heli in particular. And we again quantified them using the fish flow and the qPCR analysis, which again showed that the members of Faecalibacterium plasmodium coming down in the severe condition and showing a tendency to come back at the remission stage. And so is with the Roseveria intestinalis. But Eubacterium heli, we did not find a significant change. And this was true both by fish flow and the qPCR analysis. The decreased abundance of predominant butyrate producers like Roseveria intestinalis and Faecalibacterium plasmodium in disease condition was quite remarkable. And then comes the Lactobacillus enterococcus group. Now the members of Lactobacillus enterococcus group, what we find that in case of the moderate conditions as well as in the severe conditions, we find that there is an increase in the lactobacillus, which was a big surprise for us. So we are expecting that the lactobacilli population would come down, but maybe because of the dietary habits of Indian, population, the production of lactobacilli, even in the control samples, were higher when we compared with the literature in the European world. We find that the lactobacilli population actually goes up, and in the remission condition again we find that it is coming down to the normal flora. So this was something which was difficult for us to explain at this point of time. So the increased abundance in members of lactobacillus during disease condition was seen in our study. And these are the subdominant flora known as Atopobium cluster. And here also we find that even in the severe conditions, the population is low, but it goes up. So we don't know if all of them are pathobionts or not, but we find that there is an increase in the population of the Atopobium cluster during the disease condition. So there was a significant increase in the Atopobium during severe condition of UC. And in case of Disulpho vibrio, again we find that the population of Disulpho vibrio, that is the sulfate producing bacteria, are going high in case of moderate and severe conditions, whereas in case of remission they still maintain the level as we see in the severe condition of the disease. And this was seen both in the flow cytometry and the qPCR analysis. Now coming to the methanogens, this was very interesting that we found an increase in the Disulpho vibrio bacteria, but we found a decrease in the methanogens. The population of methanogens of course is low in a normal individual, but what we find that in the moderate and the severe conditions, the methanogens actually come down, and we specifically saw the quantitative analysis by PCR for the Methanobrevibacter smithii, and we find that in case of severe condition, we find a little high in the Methanobrevibacter species, but it was not significantly high. So the p-value was above 0.05. So there was a decrease in the methanogens during severe condition of UC. In this slide, what I show you is we made a comparative study of the firmicutes versus bacteroids. The members of the firmicutes versus bacteroids during control, moderate, severe and remission study. What we find that in case of moderate and the severe condition, the And especially in the severe condition, the ratio of firmicutes to bacteroids come down. So that means the bacteria's population go up, firmicutes go down. And during the remission, they sort of come back, revert back to the normal level. When we made a comparison of the gram-positive versus gram-negative group, what we find that the members of this ratio of gram-positive to gram-negative, that is, there is an increase, there is a decrease in the ratio of gram-positive to gram-negative because there is a change in the qPCR analysis. And in order to establish a probable link between the altered butyrate-producing bacteria and the impaired cellular metabolism, we also check the metabolites, that is the short-chain fatty acid production, using gas chromatogram profile of the fecal samples during these disease conditions. So what we did was, there is a standard, and we took the fecal samples and measured each of these metabolites. So there are, if we look into this figure, we find that there is a significant drop in the acetate production, in the propionate production, and in the butyrate production when we also measured the butyrate level in the fecal sample itself, which corroborated our observation where we saw that there was a drop in the butyrate producing bacteria. So among SCFA, butyrate and propionate concentration decreased significantly during the disease condition, and there was an increase in the lactate concentration. As I told you that we also found an increase in the lactic acid bacteria. So to prove this, what we did, can I take 2 minutes, please? So what we did was, we did a RICE study where we took a forward primer from the 16S ribosomal rRNA and a reverse primer from the 23S rRNA, and we wanted to amplify the variable region, and we took a genus-specific primer for, say, Lactobacillus. In this example, I'll just show you one example here, where we have seen that there is a severe condition and the moderate and the control and the remission. We find that if we put it on a DNHN page, we can see the number of bands which are different in these three conditions. So what we did, we took these bands and we sequenced them, and we went back to the database, and what we find that the population in the moderate, severe and remission, the bacteria which it shows matches are quite different. So similarly we did for the bacteroids, we did also for the, I'll just run through these slides, and we find that some of these bacteria are, for example, induces colitis in IL-10 deficient mice, this back one which was common in all category. So some of these bacteria could be connected with their functions as well. We did for the Clostridium leptum as well. Then we also did a phylogenetic tree of Clostridium leptum showing the sequence homology. Yeah, I'll just go through let's run through this one, please. So this is a phylogenetic tree of 16S ribosomal RNA analysis of bifidobacteria showing the sequence homology with the database. So what we concluded out of this, that since in case of the pathogenesis of the ulcerative colitis, inflammation is the central event, and what we found in our study is there is elevated levels of bacteroids which can exaggerate the mucosal antibody, and because it also, for example, Bacteroides fragilis is known to generate BFT or the enterotoxins, and in case of decreased level of butyrate producer was supported by the SCFA concentration or short-chain fatty acid concentration, which came down. There was an increased level of sulfate-reducing bacteria that is producing more of H2S, which is toxic to colonocytes and inhibits the butyrate utilization. And there was an increase in the lactobacillus, as I told you, and we found an increase in lactic acid production. There is a loss of, which can cause a loss of mucin layer, because we found a decreased expression of one of the mucin gene. So perhaps the adherence property of lactobacillus is lost because of the loss of the mucosal layer, and therefore it transits to the feces and gives us a high value of lactobacillus. This is our conclusion right now. So what we finally say that the dysbiosis can be functionally coordinated, because the dysbiosis in IBD are correspondingly not simply the structural changes in the gut microbiota, but are instead associated with major impairments in many fundamental microbial metabolic functions, with potential impact on the host. So there is a reduction in all these clades may thus have a functional consequence on the ability of the host to repair the epithelium and to regulate the inflammation. So this is my research team, and there is a poster presented by Rina who did this work, which is displayed on the poster session. Thank you very much.